right. My name is Hayley Paquette. I'm a graduate student in the scientific training program at the Canadian Museum of Nature. I study lichens and allied fungi, and over the next 4 minutes and 45 seconds, I'd like to tell you about my research. So, what is a lichen? A lichen is a composite organism. It consists of a mycobiont or fungus and at least one photobiont, which can be an algae or cyanobacteria or both. The mycobiont is responsible for the lichen's structure, and the photobiont photosynthesizes energy. This energy production that the photobiont makes possible is really cool, because typically fungi need to obtain their energy from organic matter in the environment, but this partnership with the photobiont frees them of this restriction. Lichens live pretty much anywhere where there's enough light, water, and clean air. They exist in virtually every landscape on Earth, but they especially thrive in areas where little else does. For example, lichens dominate the landscape in the Arctic. Lichens are obviously fascinating, but why care? Lichens are bioindicators, which means they can tell us a lot about the health of an ecosystem. Baseline data on lichen diversity for a region allows changes in those lichen communities to be monitored, which can alert us to changes in things like air quality, biodiversity, and forest health before they become obvious. My project looks at lichen diversity in Forillon National Park on the Gaspé Peninsula in Quebec. Forillon is special for a number of reasons, the most interesting to me being that it is the continental terminus of the Appalachian Mountain Range, which stretches across eastern North America from Alabama in the United States to Newfoundland in Canada. In Forillon, I'm conducting a floristic study of the lichens to establish baseline lichen diversity data for this area, a critical step in utilizing lichens as bioindicators. Lichens have been well studied in the southern Appalachians, but in the northern Appalachians, where my study is located, lichen diversity data is limited. That's something I really like about my study. I get to contribute understanding to a relatively understudied area. So what does all this look like before it becomes a tidy paper in a journal? My project began with an exploratory trip to Forillon and ultimately months of planning to prepare for a more lengthy exploration of every major habitat in the park and as many microhabitats as possible in search of lichens. Forillon has a surprisingly large diversity of habitats within its small area. There are maritime, boreal, and temperate habitats, all within its 245 square kilometers of terrestrial boundaries. One particularly remarkable occurrence is a taiga habitat, which is not typically seen at this low altitude. Within these dominant habitats, I also investigated microhabitats, for example, caverns of large old maple trees, cedars in wet forests or swamps, and exposed limestone cliffs. These habitats, they're a little harder to map out ahead of time. You really have to search for them once you're there. Altogether, I spent 50 days in Forillon, collected 893 lichen samples, took 168 GPS points, walked or bicycled over 300 kilometers of trails in Forillon, and ate way more Cliff Bars than I'm willing to give a number to in this presentation. And I loved every minute of it. I think this is an important component of good research. Forillon became my home. Discovering each unique habitat and thoroughly documenting the lichens living there is not just a project to pass or fail. I care that the lichens of Forillon are accurately represented in my study. During fieldwork, my office was a picnic table at my campsite, and my lab was deep within the park, far from roads, trails, and campgrounds. For me, the time I spent in the field connects me to the time that I spend in the lab and writing back in the city. Then the big switch, from field to lab. In the lab, I rediscover each collection, and I'm transported back to that moment in the field where I collected that specimen. In the lab, I get to fill in the blanks and start to really understand what is going on. I look at the unique details of each specimen, going from the 10 times magnification of my hand lens in the field up to 1,000 or more times magnification through the microscopes in the lab to examine the hidden details. My list of species is growing and new and interesting discoveries are starting to be revealed. One of those interesting discoveries is an allied fungi called Kinethicopsis oregana, or resin whiskers. This tiny species grows on the resin of conifers, and its stalks are black and shiny and about 1 millimeter tall if we're rounding up. This is the first collection of this species in northeastern North America. Prior to this, it was collected only once in Canada, in Alberta in 2013, and in the greater picture of North America, it's been collected 20 times along the west coast of the United States. But it's speculated that it's maybe more widespread, and my finding in Forillon contributes one more data point to addressing this hypothesis. So, I still have a lot of specimens to identify, not to mention writing a thesis and all the parts that go along with that. It makes a big difference to see people that look like you succeed when you are trying that look like... It's really nice to see women doing science. When things get hard, that really helps. So thank you to all of the women in science, past and present, who have succeeded and made space for me to be where I am today, discovering something I love and marking all of my field career with hot pink duct tape.